Welcome to the Education Session 10, Pi3K, or Picking Your Target. And my name is Cynthia Ma. I'm from Washington University School of Medicine, and it's my pleasure to moderate today's session. We have a great panel of speakers, and they're experts in studying cancer biology, breast cancer, Pi3 kinase pathway. And with the recent approval of capivasertib and our previous approval of Pi3 kinase inhibitors and mTOR inhibitors, I think this is a really timely session. So the hope of this session is that we'll provide you a scientific background why we're targeting this pathway and why we choose these targets, and the challenges and also success that has been there, and also how to best improve the therapeutic window of these agents that target these pathways, and also potential like future combination strategies, and what we are looking ahead. So without further ado, I'd like to invite our first speaker, Dr. Neil Vasan. He's a physician scientist at Columbia University Irving Medical Center. So he's going to talk about Pi3 kinase. Also, just to make everybody aware that we're going to save all the questions until all three speakers have speak, because a lot of the concepts and also the contents are connected. So we will have 30 minutes talk for each of the speaker, and then we will have 20 minutes question and answer. Thank you. Dr. Vasan.
Thank you. Good afternoon. These are my disclosures. So advances in the last 40 years in basic biology, drug development, understanding patient toxicities, and translational biomarkers have resulted in what I would like to term a therapeutic symmetry in how we target the Pi3 kinase pathway in 2023. I'm going to be focusing, of course, on Pi3 kinase for my talk, but certainly these drugs can be challenging to administer in the real world, and they are not, unfortunately, associated with durable responses. So it's really important for us to understand these points so that we can make advances in our field. I'll be talking about signaling and oncogenesis, how we currently inhibit Pi3 kinase in cancer, mechanisms that limit the efficacy of these drugs, how we can increase the therapeutic window with a focus on discussing mutant-selective Pi3 kinase inhibitors, and the translational future of this field. PIK3CA is the gene that codes for P110 alpha, which is the catalytic subunit of the Pi3 kinase alpha protein complex. This is shown here in green. There are 4 different isoforms of P110. Alpha is the most germane in terms of breast cancer and most cancer types. P110 binds to the P85 non-catalytic subunit, and there are many different splice variants and isoforms of the P85 subunit. There are 2 main inputs into the Pi3 kinase pathway, and those are shown here. The first is receptor tyrosine kinases, which dimerize and autophosphorylate each other, and these phosphorylated proteins bind to the P85 subunit and stimulate Pi3 kinase activity. The second mechanism is actually through RAS and other small GTPases, which can directly bind to P110 alpha and stimulate catalytic activity. As we know, Pi3 kinase is a lipid kinase that catalyzes the phosphorylation of PIP2, PI45P2 to PIP3. PIP3 is a very potent and in abundant second messenger molecule at the plasma membrane, and PIP3 recruits many proteins containing pH domains. Again, the most germane for this audience is AKT, but there are other proteins as well, and these set off intracellular signaling cascades that can potentiate many processes in cancer and other meaningful biological phenotypes. One of the most important genomic discoveries, I think, in the last 10, 15 years has been that PIK3CA is mutated in a large number of cancer types, and most notably in about 40% of ER-positive breast cancer. And these mutations occur at characteristic sites. E542K and E545K are located in the helical domain, and H1047R in the kinase domain. As you can see, there are a large number of mutations that are scattered throughout the gene, and the physiologic significance of these are poorly understood. Advances in structural biology over the decades have shown how both 110 and 85 interact with each other and how they're positioned with respect to the cell membrane. So this is a crystal structure from 2007 from Mario Emsel's group at Hopkins, which showed a full-length P110 alpha bound to the NISH2 domains of P85. That's shown in the red dotted lines here. And this structure shows extensive domain-domain interactions within the complex. And I've shown here E545 in a red dot and H1047 in a green dot. E545 is located at the interface between 110 and 85 and mediates disengaging of these proteins, leading to increased kinase activity, while H1047 is opposed close to the, within the plasma membrane binding surface, and mutation to an arginine here increases lipid binding. Interestingly, in the last couple of years, there's been extensive investigation into the structure of the full-length Pi3 kinase, and this is work spearheaded by Peter Vogt's group. This is a structure of the full-length P110 bound to full-length P85. And what's interesting about this structure, I think, is that while the resolution was not sufficiently strong enough to thread all of the domains of P80, all the extra domains of P85, what it clearly showed is that when Pi3 kinase is bound to alpelisib, BYL719, it actually induces stabilization of the complex. So you see more electron density when alpelisib is added. And this mechanism of inhibitor-induced stabilization, I think, is a really underappreciated area of the field that may spawn further drug development. Functionally, PIK3CA mutations are a weak oncogene. I'm going to just present some counterpoints here to this claim. PIK3CA mutations are sufficient to increase cell signaling and growth, but in the vast majority of cell types, growth factors have to be removed from the media to elicit these changes. PIK3CA mutations are sufficient to cause tumors in mice in genetically engineered models, but there's a very long latency period, generally a year, and the histologies that result are rarely adenocarcinoma and are usually mixed histologies with sarcomas and other lineage-dependent histologies. Pi3 kinase inhibitors do inhibit cell and tumor proliferation, but these are cytostatic mechanisms primarily, resulting in cell cycle arrest and are not cytotoxic. And finally, Pi3 kinase inhibitor combinations have clinical efficacy in cancer, but at least in the cancer, in oncology, we have no clinical efficacy to date as a monotherapy. The drug development of the Pi3 kinase field has undergone a dramatic increase in specificity of the target over the decades. And with this increase in specificity, we've also seen increased appreciation for biomarkers. So one, in this figure, drugs that are under accelerated approval or FDA approval are shown in blue. And so one drug, an important drug in the field that showed that PIK3CA mutation predicts for Pi3 kinase inhibitor efficacy was buparlisib, which is a pan-Pi3 kinase inhibitor. This was tested in the BEL-2 trial in ER-positive metastatic breast cancer, and what this trial clearly showed was that PIK3CA mutations predict for response. Now, this drug belongs to a genre of clinical trials in a sense that they are statistically significant, but clinically, unfortunately, not significant due to significant toxicities. And this is a theme that we've seen in the Pi3 kinase field for a long time. In 2019, of course, we had the report of alpelisib in the SOLAR-1 trial, which was the registrational trial that showed its efficacy in combination with fulvestrant. This was in a CDK4/6 inhibitor naive population. More recently, we had a report of the BYLieve trial, which was the single-arm phase 2 trial showing a median PFS of 7.3 months after a CDK4/6 inhibitor. Both of these trials are very important in the field, and especially the 7.3 month I think of as a real benchmark in how we think about cross-trial comparisons. We know that about 1/3 of patients exhibit grade 3 or greater hyperglycemia, and so this drug and other drugs in the pathway can be very challenging to administer in the real world. So in terms of resistance mechanisms to Pi3 kinase inhibitors, researchers in the field have really relied on cell line analysis and patient sample analysis to discover new resistance mechanisms. And these have impinged on 4 main classes of mechanisms: DNA-based mutational mechanisms, different types of pathway rewiring, effects on gene expression, both transcriptional and epigenetic, and effects on metabolism. And I think this makes sense in a way given Pi3 kinase well-known role in both mitogenesis, in gene expression, and in metabolism. And again, it's also a field where, unfortunately, we don't have great mouse models, but yet, despite these inadequacies, many of the discoveries that are made in cell lines are actually able to inform phase 1 clinical trials. So the first report of Pi3 kinase inhibitor resistance was a report of an exceptional responder patient. This was a patient a breast cancer patient who responded to alpelisib monotherapy, but then passed away. And she was found to have different PTEN mutations, convergent PTEN mutations in 10 metastases. And in subsequent preclinical work, knockdown of PTEN caused alpelisib resistance, but also maintained sensitivity to buparlisib, a pan-Pi3 kinase inhibitor. So this is an N of 1. More recently, we've seen 2 studies of similar sizes, which have again confirmed that acquired PTEN mutations are a mechanism of resistance in about 20 to 25% of patients receiving Pi3 kinase inhibitors. Interestingly, a recent report also showed acquired AKT1 mutations and acquired second site mutations in PIK3CA. And so biologically, this is, of course, very plausible, but I think we need larger numbers of studies of patients who've received alpelisib and then progressed to really understand the full spectrum of resistance mechanisms at the genomic level. There can be significant pathway rewiring that can also drive Pi3 kinase inhibitor resistance. And I've outlined this here. These pathway rewirings fall into 2 main classes. The first is AKT-independent activation of mTORC1. And there are many mechanisms that have been elucidated that can fall into this category. Activation of SGK, which is a pH domain-containing protein, overexpression of PIM, loss of mTORC1 negative regulators, and loss of INPP4B, which Dr. Toker has published extensively on. The second mechanism is overexpression of upstream RTKs, and often this happens, this is a translational mechanism. So one of the challenges here is that while there have been many, many reports that have impinged these 2 pathways and implicated these pathways, most of these genes are not represented in targeted NGS platforms. And also, since this RTK overexpression is a translational mechanism, we don't necessarily get that information from DNA-based sequencing reports. And so this is a big challenge in the field because we have many patients who are undergoing these therapies and we don't have the optimal biomarker-based platforms to fully understand the spectrum of resistance that can happen. Oh, and I'm sorry, and we also do know that, unfortunately, therapeutic targeting, co-targeting of these mechanisms has not resulted in successful combinations. This may just be due to toxicities, and I think there's more to come here. We know that Pi3 kinase is a very important regulator of gene expression, and one of the most important discoveries in the Pi3 kinase field in the last decade has been the recognition that Pi3 kinase inhibition can increase ER-dependent signaling and ER-dependent transcription through both transcriptional and epigenetic mechanisms. And here, this is showing that Pi3 kinase inhibition can modulate FOXO3A, which can increase ER-dependent transcription, and can also inhibit the phosphorylation of KMT2D, which is a histone methyltransferase that can also increase ER-dependent transcription through enforcing a more open chromatin state. And so these mechanisms, I think, are very important both in breast cancer and also call into question if they may be relevant in other cancer types where we're not seeing great efficacy of these drugs. And finally, metabolic mechanisms. We know that Pi3 kinase can inhibit mutant Pi3 kinase, or excuse me, Pi3 kinase inhibitors can inhibit mutant Pi3 kinase in cancer cells, but it can also inhibit wild-type Pi3 kinase in the liver and other metabolic organs. And this can leads to increased glycogen breakdown, hyperglycemia, signaling back to the pancreas to make more insulin. And this hyperinsulinemia can actually feed back onto the cancer cell, and in preclinical models can attenuate the effects of Pi3 kinase inhibitors. And so any way that we could disrupt this feudal cycle would be predicted to increase the therapeutic window of Pi3 kinase inhibitors. And this is, I think, a really exciting area that engages both cancer biology as well as endocrinology and other areas. Some of those mechanisms to disrupt this feudal cycle are shown here. Diet, both the ketogenic diet and fasting mimicking diets have been reported to increase the therapeutic window of Pi3 kinase inhibitors. These diets obviously have a lot of effects in the body, not just effects on glycogen breakdown. The diabetic space, of course, has been transformed by many new first-in-class drugs that are non-insulin anti-hyperglycemics, and SGLT2 inhibitors and GLP-1 inhibitors are both finding very interesting roles in cancer, both with and without PI3 kinase inhibitors. And finally, I think a really underappreciated area is the concept of alternative dose schedules. So we've seen reports with PI3 kinase delta inhibitors that changing the schedule of dosing in preclinical models can actually improve the therapeutic window, decrease side effects. And of course, capivasertib is administered 4 days on, 3 days off. So that's another, I think, example where the dose scheduling may actually be contributing to a therapeutic benefit. But perhaps the most forceful way to disrupt this signaling pathway would be with a mutant-selective inhibitor, because a mutant-selective inhibitor would be predicted to not inhibit wild-type PI3 kinase and therefore not cause hyperglycemia upstream. So there are many new PI3 kinase pathway inhibitors in 2023. It's a really exciting area, and I'll discuss several of these drugs. So the first drug is inavolisib, which is an orthosteric PI3 kinase alpha inhibitor, ATP competitive inhibitor. This drug is more potent than alpelisib in preclinical models, and also has a secondary mechanism that's been shown preclinically where it also degrades P110 alpha in a dose-dependent manner. So these are Western blots in the red box of P110 alpha in cell models and in xenograft models. And this degradation is reversed by proteasome inhibitors, so it is an on-target mechanism. We know that we found out just a couple days ago that the INAVO 120 trial, which was a first-line trial testing inavolisib with palbo and fulvestrant, has met its primary endpoint of PFS, and Dr. Komal-Javeri will be presenting these data on Friday at this conference. The INAVO 121 trial is a second-line trial testing INAVO with fulvestrant versus alpelisib with fulvestrant. I'd like to point out this is an interesting trial because this is a second-line, or this is a control arm, but using a targeted therapy combination. And we're also getting some preliminary data on INAVO plus giredestrant and oral SERD at this conference. The phase 1 data of this drug showed a hyperglycemia rate, grade 3 or greater, of 22%, which I think is closer to alpelisib than some of the mutant-selective inhibitors, but of course we'll see more toxicity data. And I think one key question is really trying to understand if this degradation mechanism of P110 alpha is also true in patient samples, and if this may be contributing to a benefit. So that's an orthosteric inhibitor. What about allosteric inhibitors? So Roger Williams, a real pioneer in structural biology and the PI3 kinase pathway, had published a paper in 2012 of PI3 kinase alpha bound to PIC108, which is a tool compound. And what was surprising about this structure is that it showed 2 binding sites. The first binding site, shown at the top, is in the ATP binding pocket as expected. But there was a second binding site, and you can see the clear electron density of the small molecule, in the C-terminal hydrophobic pocket, very close to H1047, actually engaging this tryptophan residue, which is necessary for membrane binding. And so this recognition of an allosteric site has generated enthusiasm for a large number of allosteric inhibitors in this field. So the allosteric inhibitor that I think is the furthest along clinically is Relay 2608. This drug binds in an allosteric site in the kinase domain, but it's different from the ATP binding site. Relay 2608 is pan-mutant selective in both proteins and cells, and importantly does not induce hyperinsulinemia, both at early time points, 1 hour, and as well as later time points. These are the dark and light blue compared to the red, which is alpelisib. In xenograft models in both H1047R and E545K, ER-positive breast cancer, the drug decreases tumor growth, and in both as monotherapy and in combination with other breast cancer therapies. So at AACR earlier this year was presented the clinical data to date of a phase 1 clinical trial of this drug. This is an arm of patients with ER-positive metastatic breast cancer, most of whom received the combination of the Relay drug with fulvestrant, 24 patients. And this drug is associated with an overall response rate in this small phase 1 study of 17% with 4 partial responses. So, you know, I think there's many interpretations of these data beyond the fact that it's small numbers. The BYLieve trial actually also had a response rate of around 17%, and that was in a less heavily pretreated population. We also know that again, these are small numbers, and so it'll be interesting to see as this trial accrues if that overall response rate may increase. Relay 2608, importantly, is associated with on-target activity, clearing PIK3CA mutant ctDNA within one cycle, and has very little hyperglycemia, very little grade 3, and was associated with just, grade 1 and grade 2 hyperglycemia. This drug, of course, is in an ongoing clinical trial, and I think some key questions are if ctDNA clearance may predict for efficacy, and also in data that I didn't show here, this drug is associated with a flat pharmacokinetic profile, and so really wondering if the maximum tolerated dose has been reached. There are pan-mutant selective, other pan-mutant inhibitors. STX478 was also reported. Interestingly, this drug also binds in a very similar site as Relay 2608, involving many of the same amino acids. Interestingly, in the biochemical models, this drug is selective for H1047R, but in a cancer cell line context, and this is data from the Broad Prism library, that this drug actually had selectivity in both helical and kinase domain mutant cancer cells. And in many preclinical models, both in other head and neck cancer and in breast cancer, this drug had activity as monotherapy and in combination with fulvestrant and CDK4/6 inhibitors. So I think that one important question here is really, how do these mutant selective inhibitors work? Why is there this discordance between the biochemical data at the protein level and at the cellular level? And so this is something that I think we need to understand better as a class. And also, how will the clinical efficacy of these drugs differ given that they're actually binding in the same site? Another mutant selective inhibitor is LOXO783. This is an H1047R selective PI3 kinase alpha inhibitor. Again, specific for H1047R in breast cancer cell models, does not increase insulin either. And interestingly, also has activity in the brain with decreased intracranial tumor growth, and so suggesting that this drug may cross the blood-brain barrier. This drug is also in phase 1 trials in combinations and as monotherapy. We do await chemical and structural data of this compound. I think the big question here is how these H1047R specific inhibitors clinically may compare with the pan-mutant selective inhibitors. And there are two other compounds that are reporting that are at this conference, which are H1047R selective inhibitors. Another new drug in this space is gedatolisib, which is an orthosteric dual PI3 kinase mTOR inhibitor. So this is a drug class that I think really fell out of favor many years ago due to toxicities. This drug is an IV dual PI3 kinase mTOR inhibitor, and interestingly, in a phase 1 trial with intermittent dosing, 3 weeks on, 1 week off, there was a very high PFS of about 13 months in patients who had received a second CDK4/6 inhibitor, so these second, third-line patients, in populations of patients both with and without PIK3CA mutations. So this has led to this ongoing phase 3 trial called Victoria-1, testing this drug in many combinations in patients with and without PIK3CA mutations. And I'd like to point out the Arm E of this trial is also an alpelisib plus fulvestrant, so a targeted therapy combination control arm. And again, I think here one important question is the tolerability and perhaps PFS improved due to this less frequent dosing? This drug is associated with a high rate of stomatitis, 27% grade 3 or higher. That is without dexamethasone mouthwash. And so again, managing these AEs will be very important. And the last drug I'd like to tell you about is a different type of drug that binds to the RAS PI3 kinase binding interface and inhibits downstream signaling. So this is BBO-10203. This is a compound that inhibits RAS stimulation of PI3 kinase and inhibits signaling, not because it inhibits kinase activity, it doesn't, but it prevents PI3 kinase from accessing PIP2 at the plasma membrane. And so what's interesting is that this compound, when tested in many preclinical models, had efficacy in both helical domain mutated cancer cell lines, so E545K, and also HER2 amplified cell lines. And I think this is very interesting because we know from classical work from Peter Vogt's group that the helical domain mutations require RAS GTP binding for activation. So this dovetails very nicely with our basic science understanding of how these mutations function. This drug, again, does not induce hyperglycemia and has activity in xenograft models. So now I'd like to talk a little bit about the translational future of our field. We've talked a lot about resistance to these drugs, but what about sensitivity? We know that patients can have a very, very large spectrum of response, both in terms of response rate and in terms of progression-free survival. And several years ago, we reported that double PIK3CA mutations are a biomarker of increased sensitivity to PI3 kinase inhibitors. These double mutations increase kinase activity, increase PI3 kinase signaling, and increase response to PI3 kinase inhibitors, both in vitro and in patients. And we have further analyzed this phenomenon of multi-PIK3CA mutations more recently, where we found that they are present in about 9% of all PIK3CA mutant tumors and 12% of all PIK3CA mutant breast cancer. They're also prevalent in many other women's malignancies, including uterine cancer, vaginal cancer, and cervical cancer. We did an analysis into the different codons that were involved in these multi-PIK3CA mutant tumors, and this is a modified Circos plot where the amino acids 1 to 1068 are plotted on the circumference, and each chord connecting 2 points on the circumference is a double mutant combination. And so in this overall analysis, we found that codons 726 and 453 are the most frequently found in double mutations. This was in the overall cohort. But when we drill down into individual cancer types, you can just see visually there's a very, very large variation in the pattern of these double mutant combinations according to cancer type, breast, endometrial, cervical, and other. And despite this wide divergence in multi-PIK3CA mutations, we actually found, at least in breast cancer, a convergence in the response to PI3 kinase inhibition. So we didn't really see differences in response to PI3 kinase inhibitors, and this was a retrospective analysis of the Sandpiper trial testing teclisib plus fulvestrant. I mentioned at the beginning that there is a large number of PIK3CA mutations that are not located at hotspots. And this is some unpublished data in collaboration with Foundation Medicine, where using a very, very large patient sample set of 63,000 tumors, we found almost 2,700 distinct mutations in PIK3CA. And thinking about these mutations, we know there are classical mutations, 542, 545, 1047, and across cancer types, that only adds up to about 50% of tumors. And so we know that 50% of PIK3CA mutant cancers are these atypical mutations. And I think this is a real scenario that we see in the clinic often, is that patients have these rare PIK3CA mutations that are found on sequencing reports, and we don't know what to do as a community. We don't know if these mutations are activating. We don't know if they predict for drug sensitivity. And I think that this is a big problem in the field that needs to be answered. These numbers of mutations are also true in breast cancer, where the atypical mutation frequency is about 30%. And finally, in terms of, I mentioned this before, but I think we need to really broaden what our definition is of biomarkers. We've focused a lot on genomic biomarkers, and that has gotten us very far, but there are still many other mechanisms. We know PI3 kinase is critical for gene expression. We know that it's part of a signaling pathway where there's ample phosphorylation. We know that it is a critical metabolic regulator. And so we need to have better omics platforms of patient samples to really understand the gamut of resistance to PI3 kinase inhibitors. And in the just some provocative questions and answers that I'd like to throw out. Have we reached an efficacy ceiling for PI3 kinase inhibitors in ER-positive metastatic breast cancer and other cancers? And I think that we are reaching an efficacy ceiling. And I think it's really critical that we have to be testing combinations. It's very clear that many of these drugs tested in the late lines, it can be very hard to extrapolate what that means in a first-line or second-line setting. I also think that in other cancer types, we've still seen very low response rates of mutant selective inhibitors as well in other cancer types. And so we need to be aggressively testing more combinations to find more biomarkers of sensitivity. Do mutant selective PI3 kinase inhibitors solve the hyperglycemia problem? I think that we're seeing a lot of data, both from preclinical models and from clinical studies, that the answer to this is yes. Should targeted therapy or therapies of physician's choice be standard control arms in post-CDK4/6 inhibitor trials? I think the answer is yes, and this is a complicated question, a complicated answer that will have a lot of implications both for patients, oncologists, and regulators. Do atypical PIK3CA mutations predict for PI3 kinase inhibitor or AKT inhibitor response? We really don't know. And can Pi3 kinase inhibitor resistance be overcome by further inhibition of the Pi3 kinase pathway, either with AKT or mTOR? And this is unclear, and I hope we'll get more clinical answers to this in the future. So finally, I'd like to acknowledge my laboratory and my funding sources. And in the last couple of minutes, I would like to discuss a really exciting initiative that we're announcing today and launching today. PIK3CA is the most frequently altered gene in breast cancer, but we have significant gaps that prevent optimizing care in the real world. And we are very inspired by a large number of lung cancer biomarker-focused groups by our colleagues, and these patient advocacy groups have accomplished tremendous things for treatment, for interfacing with industry, enrolling patients on trials, and just raising awareness. And we don't have a PIK3CA patient advocacy group thus far. And so to address this gap in the field, we're really proud to launch a new organization called PIK3CA Pathbreakers, which is a new patient advocacy group. I'm thrilled to be partnered with 3 incredible patient advocates shown here, and they're sitting in the front row right now. And we represent a group of patients dedicated to improving outcomes for people living with PIK3CA-altered breast cancer. We have several goals in the first year. We want to develop patient-centered educational materials talking about sequencing, drugs available, and side effects. We want to create a curated clinical trials platform or portal for Pi3 kinase inhibitor trials, raise funding for translational research in this field, and organize an annual research summit. And we're really excited about partnering with patients, oncologists, representatives from industry and the government to raise more awareness for PIK3CA and for other biomarker-directed therapies in our field. These are QR codes for our website, pik3capathbreakers.org, and our Twitter account, PIK3CA Pathbreak. You can use the QR codes. Please visit our website, sign up, and help amplify our message. Thank you, and look forward to the rest of the talks.
Thank you, Dr. Vasan, for that wonderful discussion. Sorry. Thank you, Dr. Vasan, for that wonderful discussion, and also the congratulations on that PIK3CA Pathbreaker initiative. Hope it's going to generate a lot of interest. And yeah. So next, I'd like to introduce our next speaker, Dr. Alex Toker from Beth Israel Deaconess Medical Center, Harvard Medical School. He's going to talk about identifying collateral vulnerabilities in the AKT pathway for therapeutic benefit.
Thank you, Dr. Ma. Thank you to the organizers and the program committee for inviting me and giving me an opportunity to tell you about some of our work in this space. I am tasked with telling you about AKT and AKT inhibitors in breast cancer. And this really could not come at a better time because, as all of you know, 3 weeks ago to the day, in fact, almost to the hour, the FDA approved the first in-class AKT inhibitor for ER-positive breast cancer patients in combination with endocrine therapy, fulvestrant, an event that I've been waiting for since 1994 when I started working on AKT. More about this later. These are my disclosures. This is my diagrammatic view of the Pi3 kinase and AKT signaling pathway that, as Dr. Vasan highlighted, is initiated by the biosynthesis of the most negatively charged second messenger in all cells, PIP3, which transduces the signal from Pi3 kinases to activate downstream signaling signal relay molecules, of which arguably the most studied one is the serine-threonine proto-oncogene kinase, AKT. AKT transduces a signal to cellular responses through the phosphorylation of over 200 downstream substrates, which regulate virtually every single biological processes in all cells and tissues, including cellular survival, proliferation, as in DNA synthesis, cellular metabolism, cell growth as in size, and many other phenotypes, all of which are deregulated in pathophysiology such as breast cancer. Three major evolutionarily conserved multifunctional roles transduce the AKT signal, these being the transcription factors of the FOXO family, glycogen synthase kinase 3, which transduces by and large the metabolic functions of AKT, and the tuberous sclerosis complex 2, which transduces a signal to the mTOR1 pathway. As you all know, this is the most genetically deregulated pathway in all cancers, not just in breast cancer, but in all cancers. If you query TCGA for genes that participate in signal relay in this pathway at the level of somatic mutations, amplification, deep deletions in the case of tumor suppressors, the Pi3 kinase AKT mTOR signaling pathway is the most, in aggregate, genetically deregulated pathway in all of cancers. And this is especially true in women's cancers, including endometrial cancer, breast cancer, etc., ovarian cancer. But at the level of AKT, and we often talk about AKT as a single gene, but it is, as you all know, comprises 3 genes which are located on distinct chromosomes, AKT1, 2, and 3. And the level of genetic deregulation is quite distinct, whereby AKT1 is typically subjected to recurrent somatic mutations, which I'll talk about in a moment. But these are rarely, if ever, seen in the case of AKT2 and AKT3, whereby you typically see copy number gains, especially in the case of AKT3 and in the case of triple-negative breast cancer. In fact, one could argue that AKT3 is actually a driver oncogene in TNBC because of recurrent copy number gains in this molecular subtype. The first identification of a recurrent somatic mutation in AKT is the famous E17K mutation in the plextrin homology domain that was first identified by John Carpenter and James Thomas at Eli Lilly, whereby mutation at residue 17 causes an alteration which causes a genetic alteration which causes constitutive association of AKT at the plasma membrane, thereby bypassing the need for PIP3 engagement and constitutively activating the kinase, thereby leading continuous signal relay. And that is highlighted here. You do not see recurrent AKT2 or AKT3 recurrent somatic mutations at E17K. They typically tend to be sporadic or absent, but they do exist in other cancer lineages. And as Dr. Vasan mentioned, as you all know, over the last 30 years or so, there has been tremendous activity in the preclinical and clinical arena with the development of pan-Pi3 kinase inhibitors, isoform-selective Pi3 kinase inhibitors, dual Pi3 kinase and both catalytic and allosteric mTOR inhibitors that you hear about in the next presentation, and AKT inhibitors, which up until 3 weeks ago, I would continually say, have yet to be approved by regulatory agencies. And this is what I'm going to spend the rest of my talk on. So as Dr. Vasan highlighted, the alpelisib trial, the SOLAR-1 trial led by Fabrice André, was really a landmark event in the field because it catalyzed the approval of the first P110 alpha specific inhibitor in ER-positive breast cancer patients with a PIK3CA mutation in combination with fulvestrant. I just want to point out, especially for any trainees in the audience, that this came nearly 40 years after the discovery of Pi3 kinase by Luke Antley and colleagues in the mid-1980s. It took almost 40 years of research, drug development, preclinical studies, clinical trials, and regulatory approval to arrive at the approval of alpelisib. This is a partial list of some of the AKT inhibitors in preclinical and clinical development. The top are catalytic inhibitors, and the ones that I'm going to focus exclusively on in this presentation are the lead compounds, ipatasertib, developed by Genentech, Roche, and the AstraZeneca compound, capivasertib. There are, of course, many other AKT inhibitors that have been used over the past decade, some of which have not progressed in phase 1 or phase 2 clinical trials, but have served as tremendous tool compounds for discovery science, and that cannot be underestimated. Many of these are in trials in phase 1, phase 2, and even phase 3, but typically for combinations. And a few years ago, the Lotus phase 2 clinical trial combining ipatasertib, the Genentech compound, with paclitaxel in triple-negative breast cancer patients really gave us hope, and this was presented here at San Antonio, really gave us hope that this would be the first approval, or lead to the first approval of the first AKT inhibitor. And it immediately spurred the opportunity phase 3 clinical trial of the same combination, the same drugs, the same triple-negative breast cancer population, which showed absolutely no benefit whatsoever in the phase 3 trial. And unfortunately, this led to a lot of disappointment by people like me, and I dare say Genentech. But of course, I think this is still a fantastic drug, and I do believe that this will ultimately receive approval in other lineages because both ipatasertib and capivasertib are really tremendously potent and exquisitely specific catalytic AKT inhibitors. So why have these drugs fared so poorly? And this was alluded to by Dr. Vasan. We thought for a long time that these ATP competitive inhibitors would have tremendous lack of selectivity. This is not the case at all. These are tremendously, exquisitely specific drugs. Toxicity, dose-limiting toxicities, such as hyperglycemia and hyperinsulinemia, as you just heard, but others as well, such as diarrhea and rash, really limit the efficacy of these agents in the clinic. There are nodes, positive and negative feedback nodes at virtually every step in this pathway, which is why resistance mechanisms develop so rapidly in patients treated with these agents. And we have to remember that these are kinases, but they have other regulatory moieties in the molecules, which have, in some cases, been shown to have kinase-independent functions that could be relevant for pathophysiology. So armed with this information, a few years ago, I got together with my friend and colleague, Nathaniel Gray, then at the Farber, now at Stanford, to use perhaps a slightly different strategy for targeting AKT, and that is to develop degraders. And so we did that. These are PROTACs, as I'm sure you all know. They use a very different polypharmacology for inhibiting enzymes or intracellular proteins, whereby you take two different warheads, one that a ligand that engages an E3 ubiquitin ligase, another warhead that engages your protein of choice, in this case AKT, with a chemical linker of defined rigidity and structure and length. And you make the molecule, and the idea is that you put these into cells and tissues and organisms, and the E3 ligase ligand brings the target of choice into proximity to the E3 ligase and shuttles it for degradation by the 26S proteasome, so that instead of getting catalytic inhibition, you get wholesale protein degradation. And we made the very first in-class AKT degrader that is now published, and if you're interested in this, it was published in Cellchem Bio. You can scan the QR code. It'll take you straight to that PDF. And we were very happy with this because this degrader outperformed every single AKT inhibitor, catalytic or allosteric, at least in preclinical models. But we were disappointed with relatively slow degradation kinetics, and so we set about making a second generation AKT degrader, and that's what I'm going to show you. Now, I can only show you a few slides in the interest of time. This is now in press, and hopefully it will be coming out soon. On the left, you see our second generation degrader, which degrades all three AKT isoforms because the warhead that we used for this degrader is GDC0068, ipatasertib. And you can see that at low nanomolar concentrations and over time, at 100 nanomolar or less, and over time, it is a much more rapid degrader at degrading AKT compared to our first generation degrader, which is on the right-hand side. So within a few hours, you get wholesale degradation of AKT and inhibition of downstream signaling. And when you profile the activity of these degraders, both the first and second generation, in vitro assays in cells, these are T47D ER positive, HER2 negative, PIK3CA mutant cell lines, and all you really have to pay attention to are the curves on the, let's see if I can find the cursor. The curves in black and orange, these are our first and second generation degraders, which outperform every single catalytic and allosteric AKT inhibitor, at least in vitro. And so we then initiated a collaboration with AstraZeneca, which is ongoing, who used, and this is just one of the data slides, who used their Horizon cell line profiling to profile thousands of cell lines across dozens of different lineages with distinct, with full genome annotation. And all that you really need to know about this slide is that anything below the black line shows that this particular lineage, which is color-coded, is more sensitive to INY05040, which is our second generation AKT degrader, than it is to the parent catalytic inhibitor, GDC0068. And it turns out that all of the lineages, all of the cell lines that belong to lineages whereby the degrader outperforms the catalytic inhibitor, do so because those are oncogene addicted to Pi3 kinase AKT, because they either have copy number gains of AKT, somatic mutations of AKT, PIK3CA mutation, or copy number gains, PTEN inactivation, etc., etc. And I only have time to show you one slide on this. These degraders have activity in vivo. They degrade AKT in xenograft experiments. And in terms of efficacy, this will have to wait for another presentation. What I want to really turn to now is the fact that we now have an AKT inhibitor in the clinic. As I just said, 3 weeks ago, the FDA approved capivasertib in ER-positive breast cancer patients in combination with endocrine therapy as a result of the Capitello 291 study that was led by Nick Turner and colleagues that was actually presented here at San Antonio about a year ago and was subsequently published in the New England Journal a few months ago. That showed a significant increase in progression-free survival compared to placebo with Capiva plus fulvestrant. And the most remarkable thing that I think we could perhaps later spend some time discussing is that AKT pathway mutations, which include PIK3CA, which include PTEN inactivation, which include AKT1 E17K, were not necessary to generate a response. This, to me, was extremely surprising. And I anticipated that the FDA would approve this combination for all comers. But in fact, the FDA approved this combination only in pathway-altered ER-positive breast cancer patients. That is to say, those with PIK3CA, AKT1, E17K, etc. Now, of course, there is still time in the future, depending on how this combination therapy does in the real world. There is hopefully still time for the FDA to revise this indication. But this is really tremendously exciting. And what this tells you is that if you know what the collateral vulnerabilities are in a particular lineage, you can develop combination therapies that are going to have a therapeutic window of efficacy. So how do we go about that? So for the rest of the talk, I could have talked about all the AKT trials that failed, or all of the AKT trials that are ongoing, and which we hope will give us successful combinations. But instead of doing that, I'm going to tell you our efforts, our unpublished efforts at identifying such combinations. And I just want to point out for the trainees and the audience, it's a very similar story to Pi3 kinase. 3 weeks ago, capivasertib was approved. The first publication in which the cloning of AKT was reported was in July of 1987 by Stephen Stahl in this PNAS paper. The cloning of AKT1 and 2 from a primary human gastric adenocarcinoma. Now, I wonder how many people in the audience know what AKT stands for. When I started as a trainee in the early 90s, there were 4 papers in PubMed on AKT, essentially the cloning from various sources. There are now over 100,000. Now, maybe you don't care, or maybe I'm the only person in the room who cares what AKT stands for. Maybe I'm the only person in the world who cares what AKT stands for, but I'm still going to tell you. It is a gene that is encoded that was identified from a transforming retrovirus isolated from a thymoma cell line known as AKT8. Now, that's just number 8 of that series of cell lines, but that doesn't tell you where the name comes from. The name comes from a stock A, strain K, from a mouse, originally inbred in the laboratory of Rhodes and Rose at Rockefeller. And that's where the name AKT comes from. So stock A, strain K, and then the question is, where does the T come from? And this has probably been lost in the sands of time, but it most likely stands for thymoma, because it was a mouse thymoma, or transforming, because it was isolated from a transforming retrovirus. So just a bit of info for you, if you ever wondered. Just to point out that it took 36.4 years, or 436 months and 2 weeks, since the discovery, the cloning AKT, to the approval of capivasertib. This is really remarkable. Okay, so just to go back to what I was saying, if we can identify collateral vulnerabilities in cancer lineages to these amazing drugs, can we develop the right combination therapies for these lineages? And what we do in my laboratory, or at least one of the things that we do, is use screens to identify such vulnerabilities. And the lineage that I'm going to tell you about today is triple-negative breast cancer, which, as you know, has a very high AKT gene signature, not because of overt PIK3CA mutations, although they do exist, but because this is a tumor suppressor loss disease. In addition to P53, which is inactive in virtually every triple-negative breast cancer patient, this is a PTEN and IMPP4B-driven disease. Albeit very molecularly heterogeneous, it has a high AKT gene signature, hyperactivation of AKT because of PTEN inactivation and inactivation of the other tumor suppressor in this pathway, IMPP4B. And we have drugs that are now FDA approved. And so what we set about to do was to do a synthetic lethal negative selection screen to identify synthetic lethalities to PI3 kinase and AKT inhibitors. We took a triple-negative breast cancer cell line. This one happened to have PIK3CA, but it doesn't matter. It had hyperactivation of AKT. Transduced it with a whole genome lentiviral library that we obtained from my colleague at Harvard, Steve Elledge, and then grew up the cells and then treated them with cytostatic doses of alpelisib on the top or ipatasertib on the bottom or control vehicle. Grew these out for 72 hours, and that's really important. And then did next-gen sequencing and these were all barcoded, measured the next-gen sequencing to generate synthetic lethal genes that would drop out with the anchor drug. On the left-hand side, alpelisib. On the right-hand side, ipatasertib. And anything on the bottom, below the axis, would represent a synthetic lethal combination or a synthetic lethal hit. Anything above the axis would represent an enrichment of a gene that, when knocked out, would accelerate growth of an already rapidly growing tumor cell. And this is where you would typically see tumor suppressors. And indeed, both PTEN and TSC2 fell out on the top arm. But what we're interested in to develop combination therapy strategies is what is synthetic lethal to these two anchor drugs. And after a lot of bioinformatic analysis and these kinds of hallmark gene sets, we settled on cholesterol homeostasis. Every single gene or protein product of a gene that participates in cholesterol homeostasis, both uptake, trafficking, and localization, fell out as a synthetic lethal to the AKT inhibitor arm. And the two master regulators of the steroid response element, or the steroid response system, SREBP1 and 2, encoded by SREBF1 and 2, were synthetic lethal to ipatasertib. And one of the other reasons we chose this pathway as a potential collateral vulnerability to AKT inhibition is because there are drugs that people take every single day. Millions of people take them every single day. And these are the statins, of course. The statins are the inhibitors of the rate-limiting step in cholesterol biosynthesis, HMG-CoA reductase, and this is the pathway whereby acetyl-CoA is made into HMG-CoA, and then the rate-limiting step at the enzyme, HMG-CoA is turned into mevalonate, which fuels the biosynthesis of the prenylation substrates FPP and GGPP, which fuel both protein prenylation as well as cholesterol biosynthesis. There are many statins on the market, and I show these on the right. The main ones in the use in the United States and North America and Europe are lovastatin, rosuvastatin, and others. But there's one that we decided to settle on, and that is pitavastatin, because it has a much lower dose range in humans, much higher bioavailability, and its liver metabolization is quite distinct from the other classes of statins. And so we did synergy experiments in vitro. These are all in vitro preclinical studies where we take triple-negative breast cancer lines and ask the question, if you do synergy combination experiments to look at inhibition of proliferation, where you increase the dose of pitavastatin, do you lower the IC50 for the AKT inhibitor? And the answer is you do, for both ipatasertib on the top and capivasertib on the bottom. And this is true synergy. You can measure actual synergy. You can do synergy calculations to show you that this is not additivism, this is true synergy. This also induces cell death. These are incucide experiments measuring caspase cleavage in two triple-negative lines with a positive control in the pink being bortezomib, which the combination of capivasertib and pitavastatin induces cell death, but the two monotherapy agents are essentially completely cytostatic or do not induce cell death. Remarkably, when we expanded our screen to ER-positive cell lines, we saw absolutely no synergy whatsoever. So ER-positive breast cancer cell lines are non-responders to this combination. So these are 3 T47D, MCF7s, BT474s, and the lines are essentially superimposable for both ipatasertib on the top and capivasertib on the bottom. We also did what I thought was a cute experiment where we took T47D parental lines from my colleague Miles Brown at the Farber, who had generated fulvestrant-resistant lines, 3 independent clones. And you can see that the parental T47Ds are completely insensitive, but the fulvestrant-resistant lines have now been transformed to responders, essentially look like triple-negative breast cancer lines. And so in aggregate, when my student, Alessandra Hillis, who's done all this work, put all of these data together with multiple ER-negative and ER-positive lines, this is one of those things in biology that you rarely see. It's black and white. It's all or nothing. Every single ER-negative line that we have tested is a responder. Every single ER-positive line is a non-responder. The IC50 for AKT inhibitors simply do not shift with pitavastatin. I should point out that you see these exact results with the other statins. You just have to use much higher doses. This becomes very important later on. This is not just true in sort of simple cells growing on plastic. This is also true in patient-derived organoids. These are 3 ER-positive metastatic breast cancer organoids that we've done in collaboration with my colleague Taru Muranan at the BI. This is a single agents, capiva and pitava, or the combination, staining for EDU in red, green for cleave caspase 3. There is essentially no activity. These are non-responders. But in ER-low metastatic breast cancer lines, you see a robust induction of caspase cleavage and complete inhibition of cell growth as marked by EDU staining. So this is true not just in patient-derived cell lines, but in primary patient-derived organoids. And it's also true in mouse xenografts in vivo. It took us a year to develop these in vivo studies because we had to alter the mouse diet, the mouse chow for reasons that I don't have time to go into, but actually pertains to some of the comments that Dr. Vasan was making about the particular diet that is required to observing therapeutic windows. The combination retards tumor growth in these tumor size at endpoint and tumor percent body weight at endpoint with no effect or minimal effect of the 2 combinations at these doses. So the hypothesis that we have set up is that triple-negative breast cancer cells have deregulated canonical cholesterol sensing such that they cannot upregulate HMG-CoA reductase expression in response to low sterols. If I had another 30 minutes, I could tell you the mechanistic basis for the differentiation between ER-negative as responders and ER-positive and non-responders, but I don't. And so that'll have to wait for another time. So this has left us with these sort of musings with discovery science that highlights a potential collateral vulnerability to AKT inhibitors, at least in triple-negative breast cancer, which we combine with looking at epidemiological data that we would like to then move into clinical trials. So some of you, probably all of you, are wondering, hang on, if there was activity for statins in combination with AKT inhibitors, and AKT inhibitors have been in clinical trials now for years, we should have seen this because patients enrolled in these clinical trials take statins. And this is by no means the first time that anyone has reported anti-cancer activity for statins, but the results are all over the place. Some say there's some efficacy, some say there's no efficacy, some say there's no activity at all. And some say actually that they accelerate tumor growth. So what do you believe? Well, there's a very good reason, because this is one of the first questions that we were challenged with. Why have we not seen activity in clinical trials for statins? And the answer is actually remarkably straightforward. And it's to do with the fact that it's doses and why we picked pitavastatin in the first place. If you look at the number, if you do some basic mathematics, ask the question, how many women get breast cancer? How many women in the US take statins? And what fraction of those take statins such as simvastatin, atorvastatin, rosuvastatin, lovastatin? and the other category, which includes 3 statins, within which is pitavastatin. It turns out that only 3% of women, because pitavastatin is not prescribed in the United States, or very, very infrequently. And so it turns out that the available patient data in the United States is simply not powered to evaluate the anti-cancer efficacy of statins like pitavastatin. And this is even worse, because if you take into consideration the female pitavastatin users that are at risk of developing TNBC, 10 to 15%, it's less than 1%. So essentially, this retrospective analysis simply doesn't work because there are not enough patient numbers to do this analysis. What is required are prospective trials. So now, I don't know. We have preclinical data, we have in vivo data, we have patient-derived organoids. I don't know if combination AKT inhibitors in combination with pitavastatin are going to be effective in patients, but I'm going to find out. Just to point out that pitavastatin is primarily prescribed in Asia. It was patented in 1987, approved for medical use in 2003, and it is prescribed primarily in Japan, South Korea, and in India. It was approved here by the FDA in 2009, but it is rarely prescribed. Why? Because it costs 10 times more, because it's still on patent, than it is to prescribe one of the standard statins, which have a much lower bioavailability, much higher dose range, and would simply be too toxic to achieve anti-cancer activity in patients. Pitavastatin comes out of patent on February 2nd, 2024. Just leaves me to thank all the people who did this work. The work on the degrader was carried out in collaboration with Nathaniel Gray, by a former graduate student, Emily Erickson, who's now Blueprint Medicines. And we are continuing that work. And all of the work on the identifying synthetic lethal combinations, of which I only was able to show you one example, was carried out by this remarkably talented grad student who will soon be leaving for a postdoc. Our Twitter handle, the very generous funding sources, particularly the Ludwig Center at Harvard, who has funded all of this work. And finally, should you ever find yourself walking around or driving around Boston and you happen to see a car with this license plate, you'll know who's driving it. Thank you very much for your attention.
Thank you, Dr. Toker. And next, I'd like to introduce Dr. Robert Schneider. He's going to talk about mTOR, and he's from NYU School of Medicine. Thank you.
Okay, thank you. Thank you all for staying. I'd like to do several things in this talk. I'd like to describe why it's been so difficult to make use of mTOR inhibitors, what the confounding issues are, and then deconvolute that, because most of the issue has to do with the right combination. And if we look at the biology of mTOR, it's largely about selective translation of specific messenger RNAs. And we're just beginning to actually capture that and develop new drugs in that area. So the first part of this talk will deal with TOR, why it's been such a frustrating target, and some of the progress that's being made, so there is hope. And the second part will actually include some preclinical data, which shows that the next horizon is to move downstream of mTOR and to begin to selectively inhibit specific elements of the translation apparatus. And one thing I have to say is that since COVID, the whole world knows that mRNA is actually translated. Even taxi drivers in New York City, when I go to work, they used to ask me when I'd go to NYU, where are you going? And say, NYU, what do you do? Breast cancer. And that was the end of the conversation. Now they say, what do you do? And I say, I work on mRNA. And they're, you work on mRNA? So they're very excited. So mRNA has finally reached a point of real recognition. Okay, those are my disclosure information. So without going into too much depth, on the left of this slide shows you why it's so difficult to drug mTOR. Because on the left and on the right, you realize there are two mTOR complexes. In fact, there are now three, and I'll talk a little bit about that at the very end. So mTORC1 actually drives protein synthesis. That's its major role. And it does so through a protein that I'll refer to quite a bit in the course of this lecture, known as the 4E or EIF4E binding protein. And so it drives the majority of mRNA translation within the cell. In addition, mTORC1 is required for cell proliferation and for ribosome biogenesis. And mTORC2 is required for cell survival and for cell mobility. And so mTOR actually hits a number of major pathways, whether they're metabolic or lipid production or protein synthesis. And so inhibiting mTOR, therefore, has a large number of side effects. And that's something we need to keep in mind as we move on to try to build better generation mTOR inhibitors. And so I'll focus largely on the major function of mTOR, which is that of protein synthesis. One of the problems with just inhibiting mTORC1, and that is what we do with rapalogs, which are the only approved mTOR inhibitors to date, is that it sets us up a path to activate mTORC2 and ultimately AKT. And so, for example, the inhibition of TORC1 de-represses a negative regulatory loop that leads to the activation of IRS, which leads to the activation of Pi3 kinase and ultimately to AKT. And so as a result, there's been a lot of activity in the development of what are known as dual mTORC1 and dual mTORC2 inhibitors in order to shut down the activation of AKT, or dual inhibitors of mTORC1 and Pi3 kinase. And so there's been a lot of activity in this area. You probably know of quite a few of these drugs. On the lower right, you can see that we have a number of mTORC1 inhibitors. These inhibitors tend to be allosteric, almost all of them, which means they're actually working upstream of mTOR to block mTOR activity. mTOR actually has to go through a conformational change to become activated. And there are the competitive inhibitors for ATP, which are actually catalytic inhibitors or kinase inhibitors, and those are shown as well. Unfortunately, none of those are yet FDA approved. It has been more than 20 years since we've had the approval of a new mTOR inhibitor. And so the mTORC1 inhibitors are in wide use. I'll talk a bit about some of the problems we have with those and how to circumvent them. mTORC2 inhibitors have seen quite a bit of clinical development, but none have been approved. At the end of the day, all of this is about protein synthesis. And so as I'll show you in the last half of this talk, we need to really focus on the best synergies of mTOR inhibitors, which is to take advantage of what we know to be selective translation of oncogenic messenger RNAs. And in fact, that's the major function of mTOR hyperactivation in cancer and the overexpression of its main target, EIF4E. And so I'll explain how that occurs. So we now have three classes of mTOR inhibitors. Shown on the left of this slide are the original rapalog inhibitors. They work upstream on FKBP12. We now have the catalytic inhibitors shown in red, and those inhibit the catalytic or the ATP domain. And now we have what are called rapalinks, which combine these two inhibitors as well. And yet, it has been, as I said, more than 20 years that we've seen new FDA approval, and that was based largely on the Bolero trial, which combined everolimus with exemestane in ER-positive breast cancer. So what's the problem here? And the problem has a lot to do with the combination of TOR inhibitors that did not make use of the biology of mTOR. So shown in the top is the Octopus trial. It's a combination trial with paclitaxel and a dual TOR inhibitor. But if you think about it, this was doomed for failure because what taxanes do is to block cell proliferation, and in part in doing that, what they're doing is driving down mTOR activity. So all we did was combine two mTOR inhibitors. On the other hand, the Victoria trial, which actually did reach its endpoint in ER-positive breast cancer, combined with anastrozole. And so what we're doing here is we're blocking the effects of an ER, of the estrogen receptor, and driving down its targets at the level of mRNA translation. So for the most part, the first take-home message from this talk is that we need to find the synergistic interactions between mTOR inhibitors and their biology. And that actually provides a future for the continued development of particularly the dual mTORC1 and mTORC2 inhibitors. They do have a home as single agents, and that, for example, is found in the CC223 trial, and that's non-pancreatic neuroendocrine tumors, which we know to be largely driven by the overexpression and hyperactivation of mTOR. In contrast, above that was a phase 2 trial, and it was a rapalog-resistant trial in pancreatic cancers, where we didn't expect to see much activity because they're not generally mTOR-driven as well. So again, the message here is it's all about combinations. There are toxicities. We know many of those, but one of the ones I want to point out is not a toxicity, but it's a real problem that we don't actually often consider in the clinic, is that the major function of an mTORC1 inhibitor is to actually increase the levels of regulatory T cells. That's how they were first actually FDA approved for graft versus host disease. So the induction of Tregs is actually about 4-fold. And of course, that's the bane of our existence in breast cancer because they're providing for immune suppression. The dual TORC1, TORC2 inhibitors do not do that. And the other major issues, as you probably know, are particularly hyperglycemia and stomatitis as well. There are resistance mechanisms, and the resistance mechanisms of both the TORC1 and TORC1/2 inhibitors, again, call home the major issue, which is that the resistance to these inhibitors is driven through the upregulation of mRNA translation mechanisms, particularly the key target of mTORC1, which is eIF4E, the mRNA cap binding protein, which is what actually brings ribosomes to the messenger RNA. And so certainly for the allosteric inhibitors, that's one of the key mechanisms. Certainly it's true for the TORC1, TORC2 inhibitors. And in fact, the mechanism, a key mechanism by which most genotoxic chemotherapies arise is through the increased expression of eIF4E that promotes the specific translation of mRNAs that are involved in DNA damage responses and cell survival. And I'll show you some evidence for that. So resistance to the TOR inhibitors is actually largely leading back to eIF4E and its selective translation of specific mRNAs that drive oncogenesis and survival. So why don't we just move downstream? And that's really what the rest of this talk is about. We can drug selectively the translation machinery. And so if you look at this pathway, what you see is that almost every oncogenic signaling pathway ultimately leads to mTOR and to the inhibition of mTOR, which causes the activation of a protein known as 4E-BP1, which sequesters eIF4E. And so we might as well then just target eIF4E, and a number of companies are now doing that. What's the conceptual basis for this targeting? It is that messenger RNAs have different requirements for the translation machinery. So the typical messenger RNA in the cell does not require translation machinery to a large extent for translation. They tend to be fairly unstructured messenger RNAs, whereas oncogenic mRNAs, mRNAs that are involved in DNA damage responses and in survival, are highly structured. And shown here on the right of this slide, in the red box, is actually the 5' non-coding region. So the translation machinery has a very difficult time translating mRNAs that are involved in oncogenesis. And of course, nature designed these mRNAs to be difficult to translate. So the major impact then of an mTOR inhibitor is to actually inhibit the translation selectively of mRNAs that are involved in transformation, in DNA damage, DNA repair responses, and in survival of cancer cells. And this is just showing you there are a number of review articles from many different groups, including my own, and a lot of research over the last 30 years that have shown that the high levels of eIF4E that we see in cancer cells, the high level of mTORC1 activity, which predominates in cancer cells, is largely involved in driving the selective translation of metalloproteinases that are involved in invasion, in specific oncogenes, in survival proteins, DNA damage responses, including PARP, in a number of other cell proliferation activities as well. And this just shows you, again, it's from a review of ours, that there's been a lot of activity in drugging the translation machinery. I'll specifically point out that much of that activity has been directed to eIF4E, including antisense oligonucleotides. And there'll be a poster here by the Ribometrics group, also on their novel eIF4E small molecule inhibitor. I think that's tomorrow. So how do we combine these? Well, it's based on what we know about mTOR. So for example, and this is all from preclinical data, but easily translated to the clinic, EGFR inhibitors work extremely well synergistically if we actually inhibit TORC1 or we inhibit EIF4E with small molecules, which we can do preclinically. Cytotoxic chemotherapies, because we can then make use of the DNA damage, and by blocking either TORC1 or EIF4E, actually increases the damage to cells because it can't repair. The same is true for anti-angiogenic agents. The same is true for anti-estrogens, that it actually blocks the resistance mechanisms. Clearly with ionizing radiation, which is actually an excellent way to introduce these agents because we then have only one systemic therapy. And now there's quite a bit of data showing that blocking TORC1 or using an EIF4E inhibitor actually synergizes with the CDK4/6 inhibitors as well. So these are all good paths forward clinically. There are clinical trials. I'm not going through all of them. I won't talk about all the failures, and I'll talk only about a few of the ones that are ongoing. In the early days, Ionis made an antisense oligonucleotide to EIF4E. They took that all the way into phase 2 trials, originally with Eli Lilly. And what they found out was that they could actually suppress EIF4E expression. It was a dose escalation study. They did not see an objective response. However, roughly half of all progressing patients were in stable disease for over 20 weeks. The major issue is it's an antisense oligonucleotide, very hard to get it to penetrate tumors with small molecules who can do much better. There's a poster here on an EIF4A inhibitor. That's a helicase that works within this complex from Effector Therapeutics. I don't know the results of that, and I think that's being presented tomorrow. The other issue, which I would like to spend the rest of the time addressing, is that the translation machinery offers us another entire set of targets. Part of the reason why mTOR inhibitors actually have not proven to be terribly effective in the clinic is because there's an alternate mechanism to translate mRNAs, and those are mRNAs that are particularly involved in metastasis cell migration, and as it turns out, that same mechanism is involved in creating T regulatory cells. And so I'll spend some time talking about it. It's based largely on work published from my group, some of it quite recently. And you can see this both in patients and you can see this in animal studies, that if we inhibit EIF4E, if we hit cells with the highest levels of mTORC1 that we're allowed to use, particularly in animals, we still have 20% or more of protein synthesis that is sustained. And it has very little effect. It has some effect on metastasis, but most of the effect is on primary tumor growth. So what's going on? What's going on is that there's an alternate mechanism of translation that's dedicated to when mTOR gets downregulated, whether it's through an mTOR inhibitor or naturally for cells that are under stress. And that utilizes a protein known as DAP5 in the gene sense EIF4G2, as shown here. And this is actually a protein that assembles a translation complex in the absence of mTOR and in the absence of EIF4E. So the work that I'll show you was carried out in my lab by 3 very talented postdocs, Amadina Lard, Olga Katsara, and Tiffany Rios. And it was published quite recently. And what they found is that this alternate complex takes over when mTOR is inhibited. So when we use mTOR inhibitors, we're actually activating a compensatory translation mechanism, and we need to target that mechanism. And if you look at patients, this is triple-negative breast cancers, and this is metastasis-free survival at the mRNA level. Patients that have high levels of this EIF4G2 mRNA do very, very poorly. And at the protein level, we see the same thing. Patients with high levels of EIF4G2, both in primary tumor and in their matched metastases, do very poorly in metastasis-free survival, and that's shown here. And as it turns out, it is corresponding to roughly 20% or so of all of mRNA translation. So it's a widespread alternate mechanism that is actually induced when we inhibit mTOR. And we found that, in fact, regulatory T cells use this mechanism. So one of the key questions in T cell biology for the last 20 years is how T regs actually are able to differentiate when they downregulate mTORC1 as a component of their lineage determination. And it turns out it's the same mechanism. So if we inhibit this mechanism, this DAP5 EIF3D mechanism, that means that we can target simultaneously the metastasis of cancers, as well as the regulatory T cells that are suppressing the immune response to those tumors at the same time. And I'll show you evidence, at least preclinically, that this works, and the evidence that we're now developing small molecule inhibitors to this as well. So again, DAP5 silencing impairs, as I'll show you, the epithelial to mesenchymal transition, the translation of those mRNAs, the translation of metastasis mRNAs, and the translation of survival messenger RNAs. I won't take you through all of this data. It's been done in both mouse and in human. I should point out that the T regulatory cell data was all done in human. So in my group, we like to start with humans, and then we make a mouse that looks like a person. And so the Treg data was all done with 80 donors in which we actually induced their Tregs. And we now will be publishing a mouse data on that as well. The major mRNAs that require this mechanism for their translation are involved in cell migration, in cell movement, in inhibiting cell death, and also in which defines the mesenchymal transition. If you were to go to a textbook and pull out the mRNAs that actually drive metastasis and the mesenchymal transition, you would come up with a group that looks like this. These are mRNAs that are all dependent upon this novel DAP5 mechanism for their translation. They're all independent of TORC1, and they're independent of the EIF4E. It includes all of the key integrins, which we know to drive metastasis. So these are all the integrins, and they are translationally induced through the DAP5 3D mechanism. And so roughly 65% of all the mRNAs that are dependent on this novel translation mechanism are actually involved in the functions of the mesenchymal transition, metastasis, cell invasion, and also inhibition of cell death. The animal studies, I'm not going to show you too many, just several of those. The important point is that the same mechanism is involved in prevention of metastasis. We can make tumor cells in which you can silence inducibly the DAP5 mRNA, as shown here. And in mice, what you see is very little effect on the primary tumor, but a really extraordinary inability of those primary tumors to metastasize, as you can see on the right. Now, of course, this is not a clinical setting. The clinical setting, of course, is does this have an impact on established metastases? And indeed it does. First, it blocks invasion. Excuse me. These cells were all labeled with red fluorescent protein. We can look at their invasion through the tumor stroma. And you can see on the upper panel that these are the wild-type cells. On the lower panel, when DAP5 is silenced, they cannot invade. There's no cell migration and no cell movement. And there's no metastasis to lung. The upper panel shown here is actually metastasis to lung in the wild type. The lower panel is when DAP5 has been silenced. It's roughly 95% inhibition of metastasis. If we implant tumors in mice, we let them grow a bit, then we take them out aseptically, and we wait for metastasis to lung. So we're no longer looking at the process of metastasis, but we're looking at the killing of established metastases. Once again, what you see is the same data. So good news as a target. DAP5 is absolutely essential, not just for the process of metastasis, but for the survival of existing metastases as well. And that's shown in the panel on the right. And again, that's the quantification of the data, roughly decreased by about 90 to 95%. As a single agent, it also increases survival, as shown here. And you can see that animals that, in which the tumors were actually silenced in DAP5 survived considerably longer, which now lends the ability for combinatorial discussion of how to position this. So we need a drug. We're working on a drug. We've actually worked out the crystal structure of DAP5. We know where it binds some of its key proteins, including EIF4A, the helicase, which is essential. And so now using virtual library screening, we have designed small molecules to fit within a druggable pocket that is unique to the DAP5 protein and not found in other family members. And that's shown right here. So I'd like to close by pointing out then that we have an excellent target to which we and hopefully others will begin to develop drugs, and that this target, this unique translation mechanism, is required for the translation of transcription factors, many of them that are involved in gene induction of transcription for metastasis and cell survival, as well as many of those factors as well. The EIF4E TORC1 mechanism is also important, and so we're in the position where we may actually have to combine either an mTORC1 inhibitor or an EIF4E inhibitor with inhibitors of this alternate mechanism as well. We have done some studies in that regard, and they're actually reasonably well tolerated. We do not make animals melt when we inhibit EIF4E. They tolerate it extremely well. And that's because fasting involves the downregulation of TORC1 and actually the downregulation of EIF4E as well. So we actually tolerate this quite well. And we think the same is true for the DAP5, which we've done as well. And so now moving forward, we need to just be biologically a lot smarter about how we use TORC1 inhibitors and TORC1, TORC2 inhibitors to take advantage of the biological synergy, which is all about selective translation, and the mRNAs that require this synergy for their translation as well. And then finally, I want to end that by pointing out there's no end to mTORC complexes. There's now mTORC3. And importantly, mTORC3 is not inhibited by rapalogs, and it doesn't involve the normal functions you would equate with mTOR, except that it utilizes the transcription factor ETS1, which is actually induced by the DAP5 mechanism I just spoke about. And we think that between the DAP5 mechanism and now mTORC3, both are responsible for a lot of the resistance that we see with mTOR inhibitors. And so we have new targets that we now need to address. I'll stop there. The groups that I'd like to very much thank, particularly the BCRF, they have supported me, I think, since I was an embryo, and they... including NIH and many others, and a number of members of my lab as well. So I thank you for your attention.
Thank you. A wonderful discussion and insight on mTOR biology. So I'm waiting for people to go to the microphone for questions. I do have a few questions from online, and the first one, maybe I'll go to Dr. Vasan. So this is in regards to PIK3R1. How does PIK3R1 mutation plays in breast cancer?
Yeah, that's a great question. So PIK3R1 mutations are rare, about 1 to 3% in breast cancer. And there have been actually many studies, preclinical studies that have looked at these mutations. There can be both mutations and stop codons. And in general, P85 mutations are thought to be, P85 is thought to be a tumor suppressor, although work from Lydia Chung, Gordon Mills has shown even neomorphic properties of these mutations. So I think the question is still out. I do think that there is enough preclinical data to support a rationale for investigating P85 mutations as a biomarker for either Pi3 kinase inhibitors or AKT inhibitors, and I think that could be a really interesting investigation in the future.
Thank you. And microphone in the middle here.
Yeah, so actually I have a couple questions for Alex. So I'm wondering, from the CRISPR screening, you showed this statin pathway is being enriched in combination with Pi3 AKT inhibitors. So what was the reason that only pivatastatin that works, but not other statin? You talk about hydrophilic, hydrophobic, but I was not quite sure about the exact difference between different statins.
Yeah, so mostly because I didn't have the time to show you, I believe I mentioned, let me just emphasize that other statins do work. They absolutely work in exactly the same way, simply by inhibiting HMG-CoA reductase. The reason we did not focus on those statins is because the doses required to achieve synergy in vitro, when you translate them to mouse or humans, would be so high, they would be prohibitively high. They would give you toxicities. They would be well above those that are taken by patients for cholesterol lowering. But if we've done those experiments in vitro and you see exactly the same synergy, you need tenfold more statins to achieve what you see with pitavastatin, simply because of its dose range.
Okay, the second question regarding the selectivity between the triple-negative breast cancer versus ER-positive breast cancer. Do you think that AKT inhibitor somehow create this reliance on a steroid or cluster pathways that make them more sensitive towards those statin drugs.
That was one of our first ideas, but it turns out that is not the answer. That is not an AKT driven. The AKT is simply causing the reason there is synergy is due to cholesterol trafficking. Okay, and that is something that is specifically induced by this combination, and for reasons that we haven't actually quite figured out yet, appears exclusive to tumors that are ER negative.
Okay, thanks.
Okay, microphone that way, and then we'll get to this one.
Thank you for the talks. As we know that Pi3K pathway is critical for immune functions, and so especially B cells and T cell functions. So my question is that how are these pathway inhibitors affecting host immunity or modifying the TME, because now the immunotherapy is coming for ER positive cancers?
So, as you know, Pi3 kinase delta and gamma are isoforms that are primarily expressed in immune cells, and many of the earlier trials that were testing less specific Pi3 kinase alpha inhibitors did have small rates of immune-like mediated side effects, colitis, pneumonitis, in some of the earlier trials. I think that the role of alpha in immunity is there is a literature on this, but it can be very hard to tease apart those effects from the effects on delta, and so I still think this is an ongoing field.
Thank you.
Okay, Dr. Atiaga.
Yeah, thank you. This is also for Alex Toker. I'm very intrigued, remain intrigued by the difference between ER positive and triple negative. So I want to ask you a question this way. If you look, if you ignore the comparison as histological subtypes, is this synergy on the basis of AKT1 mutations, PIK3CA mutations, or PTEN loss, or any combinations of those?
Any combinations of those.
Any combination of those.
Any pathway alteration, PIK3CA, we haven't looked at PIK3L1, but PIK3CA, the prevalent ones, PIK3CA, PTEN inactivation, IMPP4B loss, which is mostly epigenetically lost, and AKT somatic mutation or copy number gains in triple negative will give you sensitivity, but only in the triple negative setting. In the ER positive setting, there is absolutely no synergy, even if you have a pathway alteration.
That's fascinating. So you would see with a Pi3 kinase alpha inhibitor in triple negative and a cholesterol biosynthesis inhibitor, you would also see synergy?
Correct.
So it needs not to be an AKT inhibitor?
Correct.
Thank you.
Okay, well, we still have a lot of online questions. So one question that has a lot of points, I think a lot of people have this question, is regarding, does the AKT pathway have specific implications in those patients with ATM genetic mutation?
That's a good question. I have to say I haven't thought about it too much. So DNA repair, I guess, is the question. I have to say, when we've looked through our data sets for concurrent, sort of mutations, you typically don't see that. But I think, yeah, we just really haven't looked. I don't know if Neil has looked in the context of PIK3CA or other alterations in the pathway.
Yeah, we have not. There are several ongoing clinical trials combining Pi3 kinase inhibitors with PARP inhibitors in ovarian cancer, triple negative breast cancer as well. BRCA2, I believe, is an AKT substrate, which I think the physiologic significance of that is still poorly understood, and that could be a fascinating connection between those two pathways.
Mm-hmm. Okay, microphone there.
Yes, thank you. We think of Pi3 kinase, AKT, mTOR as the pathway, but do we have evidence that the downstream effects on protein synthesis, for example, is similar when we give these inhibitors and resistance mechanisms are similar, or are we ignoring all the other pathways that emanate off PI3 kinase, AKT, et cetera, and they have different mechanisms and different resistance pathways, et cetera.
So the answer is yes, we're ignoring it. So in fact, what we see at the protein synthesis level are very different effects. The Pi3K inhibitors are not nearly as effective and not nearly, and that's true actually for AKT to some extent, not nearly as selective in the effects that one sees. And of course, the further you get down the pathway, the less diversification, but that's still a major issue for TOR as well.
Okay, one of the online questions kind of related. Do we combine Pi3 kinase, mTOR, and or AKT inhibitors? Would they be synergistic? I guess any of you.
You know, this is getting to the idea of vertical inhibition, right, which we've seen in BRAF/MEK and other pathways. I think we probably all three would just worry about the added toxicities, but it is it's an interesting idea, and the tamping down of hyperglycemia that we're seeing with the mutant selective inhibitors, I think, make it an interesting start to start combining with those with other drugs. But one thing that's not clear is that are these still cytostatic mechanisms? If you combine cytostatic drug A and cytostatic drug B, is that still cytostatic, or might that have cytotoxicity? I don't know.
Yeah, so kind of prefer inhibiting the parallel pathways. Are you referring to? Yeah, Alex, any comments?
No, what Neil said, but I will just add that 2023 is going to turn out to be, in retrospect, in a few years from now, a really benchmark year, because we've had the allosteric inhibitors that we never thought were possible. The allele-specific inhibitors have now not only been made and are possible, but are showing some efficacy, okay? Where that will take us, I don't know. But secondly, we've had the first AKT, first-in-class AKT catalytic inhibitor approved. And so, combination strategies, I would argue, are the way to go in the future. That's where we're going to see the greatest windows of opportunity in terms of therapeutic efficacy. But I don't think we're quite done with making, 10 years ago, we'd say, well, we need better drugs. We need better drugs. We need to make better drugs. We have made better drugs, but I'm not quite sure that we're there yet. We haven't explored the ADC space, okay? Sort of putting these as payloads onto trastuzumab and other ADCs, I think, is worth exploring. Degraders, I think, hold a lot of promise. Having said that, not one single degrader has yet been approved by the FDA, although I don't think we're far off with the Arvinas, ER, and AR degraders. I'm cautiously optimistic that some of the AKT, R-AKT, and other groups have made AKT degraders will show ultimately clinical utility. Pi3 kinase, P110 degraders are going to come online. Maybe these could be turned into ADCs. So I think there is still chemical space that we can sample to increase the efficacy, but I still think that at the end of the day, combinations are going to be the way to go.
And I have to agree, with respect to the TOR inhibitors, we're dosing too high. We're knocking down protein synthesis by 50 to 70%. And the data doesn't actually suggest we need to go there at all. 25% is enough to knock down the translation of these key mRNAs that are driving DNA damage responses, for example, in survival. So there's a lot of room here for synergistic combinations, but we also have to reevaluate the doses that we're using and their limiting toxicities.
Okay, thank you. Microphone on the left. I'm sorry. Left first.
Thank you. Well, first of all, thank you for this fantastic session. I have a question for Alex. So when it comes to the results from the CRISPR screen, I believe the top hallmark that was enriched within your hits was the IL-6 JAK-STAT3 signaling. So I was wondering, considering the wealth of clinically approved JAK inhibitors, is it also a vulnerability that you want to explore, or maybe you have already explored when it comes to a potential combination partners for AKT inhibitors?
Yes. So I obviously focused in on cholesterol homeostasis, because statins were obviously the first thing that we thought of, FDA-approved drugs taken by people, easy to, lowest-hanging fruit type of, laboratory research. But there's a lot of other pathways and cell states that came out of these screens that we have mined. We first validate that was the top pathway that validated, and yes, we are following up on that. But it's very early days to be able to say anything concrete in terms of sort of therapeutic potential.
Thank you.
The other things that this has allowed us to do, because we've done many of these screens, we have Pi3 kinase anchors, Pi3 kinase inhibitor anchors, and we have AKT anchors. So you look for commonalities, okay, which of course makes the most sense, right? Because you would assume that what drops out for a Pi3 kinase would also drop out for an AKT anchor. But what it also allows you to do is to look for Pi3 kinase dependent, but AKT independent dropouts, because there's a lot of Pi3 kinase biology and pathophysiology that actually has nothing to do with AKT. And I would argue this is an immensely understudied area of work that deserves study and is likely to give us potential therapeutic approaches in the future.
Thank you. Microphone on the right.
So I have a question for Dr. Schneider. Regarding this mTOR inhibition, as you know that many of the tumor cells that are deficient of TEST1, TEST2, which induce activation of mTOR pathway, mTOR1 in particular. These tumor cells are highly sensitive to chemotherapy as well as radiation. In that sense, activation of the mTOR will sensitize to therapy, and mTOR inhibition may be counterproductive.
So I agree. Is there a question? I don't think there's any debate. I think that's actually an issue. But the dual TORC inhibitors would circumvent that. And so moving forward, most of our activity has to be in synergistic combinations with the dual TORC inhibitors.
There is a recent publication on energy communication showing that the dual mTOR inhibitors in combination with therapeutic agents can induce these residual tumors.
Mm-hmm. That's a good point. Thank you.
Yeah.
Okay, thank you. Dr. Arteaga.
Thanks for letting me squeeze one last question. It's to Neil Vasan. Neil, I think you showed data with RLY2608, the dual inhibitor. And you just remind us, is it equally inhibitory of both helical and kinase domain mutations? And if so, how can that happen structurally?
I'm sorry.
The relay drug had both H1047R and U545K.
Yes. So it does, and I agree. Dr. Arteaga, it's not clear from the structure why that would be, and I think that both Relay 2608 and the Scorpion drug, in the protein level and at the cellular level, there was more inhibition of the H1047R than the E542, 545K. And parameter that may explain that discrepancy between cancer cell lines and the protein and non-transformed cell line models is kinetic factors. So in both of those papers, the K-on rate, so the rate that the drug is bound to the protein, was much higher for the mutants than for the wild type. And so this is a big question in drug development right now, is what is the best surrogate or preclinical biochemical surrogate for Pi3 kinase inhibition when we're testing all these different mutations. And I think the idea that there might be some kinetic parameter that can be measured that can explain that is very interesting, and it was something that in both of these studies reported was a commonality. But your question is very spot on, because I think we don't understand enough about how these mutant selective inhibitors are inhibiting the different mutant Pi3 kinases, and the structures that we have. They're all crystal structures. They're not. The ideal structure to have, as you're alluding to, would be these mutant selective inhibitors bound to a mutant Pi3 kinase, and we don't have that.
Yeah, thank you.
Thank you. There's an online question for Dr. Toker. Is there synergy in the Okay, is there synergy between the pitavastatin in combination with Pi3K inhibitors?
Yeah, great question. There absolutely is. We just focused on AKT because AKT inhibitors show greater synergy and greater potency, but that just may be because of the drug's efficacy. But PI3 kinase inhibitors in combination with statins show the same exact profile and the same distinction between responders and non-responders. Absolutely.
Yeah, thank you. The other kind of similar. Have you looked at the impact of HER2 status, like in HER2 positive cancer versus negative cancer, the AKT in combination with statin inhibitors would combine?
We are doing that right now. I can tell you the one line that we've profiled does not show sensitivity, but it's an ER positive line, so all bets are off. That's something that we are looking into right now.
Okay, thank you. One last question for Dr. Vasan. So you mentioned upstream receptor tyrosine kinase, upregulated, such as insulin receptor. Would this resistant mechanism also happens to the mutation selective Pi3K inhibitors?
That's a great question. Yeah, so do the mutant selective inhibitors result in similar mechanisms of resistance as the ATP competitive inhibitors? Yeah, I don't think we know, but I would expect that they probably do. But one mechanism of resistance that's been seen with many mutant selective inhibitors in other kinases is additional mutations in the same gene, second site mutations, which is something we generally don't see with these inhibitors. So more correlative data is to come.
Okay, since we have 1 minute, I just want to squeeze in one last question. So we see PIK3CA mutation in many triple-negative breast cancers. Would you expect alpelisib or capivasertib have efficacy in these tumors?
Only in those patients which have PIK3CA mutations, but that's a relatively small patient population, somewhere between 5 and 7%. The vast majority have PTEN inactivation by multiple mechanisms or INPP4B loss by multiple mechanisms. So it would not make much sense to do trials for P110 alpha inhibitors other than in those populations. But it would have to be done prospectively.
Yes, I think before we have prospective data, we would not use them, off-label yet. I think that, thank you. I'd like to thank our speakers for the great discussion and thank the audience for staying with us. And thank you, and have a great San Antonio Symposium, the rest of your symposium days.